Welcome back to the deep dive. Today we are going to tackle something that honestly feels like it was ripped straight out of a screenplay for a summer blockbuster.
Hmm.
You know the trope. I'm talking about nanites.
Nanites, the magic word.
Exactly. Those microscopic little robots from science
Yeah.
fiction,
Yeah.
they swarm through the bloodstream, fixing broken bones in seconds or hunting down diseases, maybe even giving the hero some superhuman abilities. It is like the ultimate tech fixes biology fantasy.
Right.
I mean, I remember seeing this in movies where a guy gets shot and then this silver liquid swarms over the wound and 5 seconds later he is completely fine. But here's the thing that really stopped me in my tracks when we started looking at this stack of research you sent over.
Mhm.
We aren't talking about sci-fi anymore.
No, we aren't. And
Right,
that
right.
is the part that catches people off guard. We usually think of nanotechnology as this far-off future where we have tiny metal gears and silicon chips floating in our veins.
Right.
But the reality is actually much stranger.
And the twist here, the real twist, is that we didn't have to build these little machines from scratch using metal and silicon. It turns out nature beat us to the punch by about 3 billion years.
That is the big aha moment right there. We already have nanites. We have just been calling them by a different name this whole time. We call them viruses.
Which is a heavy word.
Right.
I mean, you say virus, and I definitely don't think helpful robot. I think of the flu. I think of pandemics. I think of my computer crashing.
Yeah.
I think of death, basically.
Yeah.
It is hard to shake that association.
It is hard, and rightfully so. Historically, viruses have been the greatest predator of humanity. But this deep dive is about flipping that script entirely. We are looking at how science is stripping these agents of death of their bad intentions and turning them into programmable machines.
Which sounds wild.
It is,
Yeah.
but to understand why this works, you have to look at what a virus actually is at a fundamental level. We tend to anthropomorphize them as these evil little monsters, but a virus isn't alive in the way a cell is. It doesn't eat, it doesn't breathe, it doesn't have thoughts. It is just a molecular courier.
A courier, like a FedEx truck.
That is the exact analogy I like to use, but let's make it even more specific. A virus is essentially a protein shell, which is called a capsid. And inside that shell is a payload.
Okay.
That payload is just information written in DNA or RNA. That is it. It is literally a USB drive inside a delivery van.
Okay, so if we stick with the van analogy, the reason we fear them is that historically the driver of that van has been a terrorist.
Exactly. The drive inside the capsid has been loaded with malicious code, instructions to hijack our cells and make more viruses until we get sick. The payload was simply replicate at all costs.
Right. But the van itself, the truck,
The truck is a marvel of engineering. It has
Mhm.
been honed by billions of years of evolution to do one thing perfectly. Navigate the body, evade the immune system, find a specific type of cell like a lung cell or a liver cell dock with it, and upload its data.
wow.
We could spend $1 trillion on nanotechnology and not build a machine as efficient as a viral capsid.
So the breakthrough isn't building a new truck, it is just swapping out the driver.
Precisely. If you strip out the death code, the part that makes you sick, and replace it with life code, instructions to repair a gene or kill a cancer cell, the virus stops being a destroyer.
It becomes a healer.
It becomes a programmable nanite.
I want to push on this a bit because it sounds simple when you say swap the code, but biology is wet and messy. It is not like drag and dropping a file on a desktop.
No, it's not.
And this leads us to the bigger philosophical shift that really runs through all the material we have today. We're moving away from thinking of biology as this mysterious organic magic and starting to treat it as a computation problem.
Yes, that is the fundamental shift of the 21st century. Biology is information, and disease is an information failure.
Let's unpack that because the numbers here are staggering. I
Oh.
was reading the notes on DNA data density and it sounds fake. It sounds like a typo.
It does, but it is real physics. DNA is the densest information storage medium in the known universe. We are talking about quaternary code, a CTG instead of binary zeros and ones.
Right. Because of how tightly that molecule is wound, 1 single gram of DNA can theoretically store over 200 petabytes of data.
200 petabytes. I looked this up to contextualize it for you listening. That is roughly the entire contents of the internet, or more than all the digital photos on Earth combined, in a teaspoon of powder.
Correct. It is unfathomable. So if our bodies are running on this incredibly dense code, then getting sick isn't just bad luck or bad humors like they thought in medieval times.
Getting sick is a software bug?
Exactly.
Okay, walk me through that. How is a tumor a software bug?
Think about what cancer actually is. It is uncontrolled growth. Every cell in your body has a program loop. It says grow, divide, check for errors, stop.
Okay.
In a cancer cell, the line of code that says stop growing now has been deleted or corrupted. The loop creates an infinite while loop in programming terms. It just keeps printing grow. That is a syntax error.
That makes total sense. What about autoimmune disorders? That is a logic error or a misclassification error. Your immune system has a database of self and non-self. In autoimmune diseases, the system's firewall is flagging self as intruder.
So it's a corrupted database entry.
Yeah.
And prions. We will get to those later, but that is even weirder.
Prions are a corrupted data type. The structure itself is wrong. But the point is, if you accept that disease is a software problem, then you have to accept the conclusion. You don't fix a software problem with a chemical sledgehammer.
Which is what a lot of drugs are.
Exactly. Chemotherapy is a chemical sledgehammer. It kills everything that grows fast. It is like nuking a city to catch a burglar. But if the problem is code, you fix it with a patch.
You send in the IT guy.
You send in a nanite carrying a software update.
Okay, so this is the setup. We are talking about reprogramming viruses to debug the human body.
Right.
And I know you listening might be thinking, okay, cool theory, sounds like something I'll see in the year 2100.
Right.
But that brings us to our first major section here. This isn't just theory, we have the receipts. This is happening now in the 2024 to 2025 timeframe.
That is right. We aren't speculating about whether this can work. It is working. We have products on the market and clinical trials wrapping up that prove the concept.
Let's talk about the workhorse of this revolution, the AAV.
Adeno-associated viruses. AAVs.
Why this specific virus? Why not use, say, the polio virus or the flu?
Safety. That is the primary reason. AAVs are naturally non-pathogenic. Almost everyone has been exposed to them at some point, and they don't make you sick.
Really?
Yeah, they don't cause a fever, they don't wreck your lungs, they are just present.
So they are stealthy.
Extremely stealthy. And they are small. They are simple. This makes them the perfect molecular courier because the immune system doesn't freak out the moment it sees them.
That makes sense.
As of the data we have here, there are 7 approved AAV products and over 110 clinical trials active.
And these aren't just for minor inconveniences. We are talking about curing blindness.
Yes. Look at Luxturna. It was approved back in 2017, which makes it the grandfather of this tech. But it is still mind-blowing. It treats a specific form of inherited blindness called Leber congenital amaurosis.
And the mechanism, how does it work?
They take an AAV, empty out its viral guts, and load it with the correct version of the RPE65 gene. Then they inject it directly into the retina. The virus delivers the code to the retinal cells. The cells read it, and they start producing the protein required to process light.
So the lights literally turn on.
For patients who were legally blind, yes. They can see stars, they can navigate a room.
That feels like magic. But you were saying it's just patching the driver.
It is maintenance.
Yeah.
Or look at Zolgensma for spinal muscular atrophy. This is a disease that used to be a death sentence for infants. Their muscles would just waste away because they were missing a gene called SMN1.
Oh.
Now we use a viral vector to replace that gene. It halts the deterioration. We are seeing kids walk who would have never walked.
So the truck, the AAV, is working. But the cargo, the payload, is getting smarter too. We have to talk about the text editor. We have to talk about CRISPR.
This is where things accelerate. If AAV is the truck, CRISPR-Cas9 is the toolkit inside the truck.
I always struggle with the acronym clustered, regularly interspaced.
Clustered, regularly interspaced, short palindromic repeats.
Yeah, let's just stick with CRISPR.
Agreed. You can just think of it as molecular scissors, but scissors implies it just cuts randomly. It is actually more like a find and replace function in a word processor.
How does it know where to cut, though? The human genome is billions of letters long.
It uses something called a guide RNA. Think of it as a GPS coordinate. You program the guide RNA to match the exact sequence of the bad gene. The Cas9 protein holds this map, floats through the nucleus, and scans the DNA.
Oh, man.
When it finds the match click, it locks on and cuts the strand.
And that cut is what fixes it.
The cut disables the bad gene. Or if you provide a template, the cell repairs the cut using the new correct code.
And the watershed moment for this was late 2023 with the approval of Casgevy. This is the cure for sickle cell disease.
This is a massive milestone.
I want to pause on sickle cell for a second because the sources go into this, and it is important to understand the human cost here. This is a horrific condition. The red blood cells deform. They turn into these sharp, sticky sickles. They get stuck in blood vessels. It causes excruciating pain. Patients say it feels like glass moving through their veins.
It leads to organ damage, strokes, and early death. And for a long time, all we could do was give them painkillers and hope for the best. We were treating the symptom.
Casgevy changes that. But the way it does it is fascinating. It's a hack, right? It doesn't actually fix the broken hemoglobin gene.
No, it doesn't. It does something cleverer. It targets a gene called BCL11A.
Okay, what does that do?
That gene is the off switch for fetal hemoglobin. See, when we are babies in the womb, we use a different kind of hemoglobin to pull oxygen from our mother's blood. It is super efficient. But after we are born, BCL11A kicks in and shuts that factory down, and we switch to adult hemoglobin.
And for sickle cell patients, that adult hemoglobin is broken.
Exactly. So Casgevy uses CRISPR to break the off switch. It smashes the BCL11A gene.
So the body just reboots the baby blood factory?
Yes. The patient starts producing fetal hemoglobin again. And fetal hemoglobin doesn't sickle. It is perfectly healthy.
That is incredibly smart. You are not trying to repair the complex error. You are just reactivating a backup system that was perfectly fine.
And the results speak for themselves. In the trials, 29 out of 31 patients were completely free of pain crises for at least a year. That is a functional cure.
But there's a catch with Casgevy. And this is the distinction we need to make for you listening. It is ex vivo.
Correct. Ex vivo means outside the living. To get this cure, you have to go to a hospital. They extract your bone marrow stem cells. They send those cells to a lab. Scientists edit them with CRISPR.
Right.
Then you, the patient, have to undergo chemotherapy to wipe out your remaining bone marrow.
Which is brutal.
It is incredibly taxing. Then they infuse the edited cells back into you and you wait for them to grow. It takes months. It costs millions. It is not a simple pill.
But the holy grail, and where the 2025 breakthroughs are pointing, is in vivo.
Exactly. In vivo means within the living. You don't take anything out. You inject the nanite, the viral vector, or the lipid nanoparticle directly into the patient, and it hunts down the problem inside the body.
This is the dream. The Star Trek hypospray.
We aren't quite at Star Trek levels, but we are getting close. Look at NTLA 2001. This is a treatment for hereditary transthyretin amyloidosis.
That is quite a mouthful.
It is a condition where the liver produces a toxic protein that folds incorrectly called amyloid. It gums up your heart and nerves. It is fatal.
And the fix for it.
A single intravenous infusion. They use lipid nanoparticles, which are synthetic cousins of viral vectors, to deliver CRISPR right to the liver. The nanoparticles get filtered by the liver naturally, so targeting is easy.
And what do they do once they're there?
They hunt down the TTR gene, making the toxic protein, and shut it off.
What kind of results are we seeing with that?
The data showed a greater than 90% reduction in that protein in the blood.
90%. That is basically turning off the tap.
It is. And then there is CTX310. This one really caught my eye in the sources because it moves beyond rare genetic diseases into the stuff that kills millions of people, heart disease.
Yeah, this was the Cleveland Clinic study from November 2025. This felt huge to me because usually gene therapy is for the unlucky few with rare conditions, but high cholesterol. That is half the population.
Right. They targeted a gene called ANGPTL3.
Biologists really need to work on their branding.
They really do. But the function is what matters. This gene regulates lipid metabolism. If you turn it off, your bad cholesterol and triglycerides drop. They found that a single infusion reduced bad LDL cholesterol by about 50%.
One shot.
One shot. Permanent reduction.
That is just, I mean, people take statins every day for decades to get those kinds of numbers. And statins have side effects. You have to remember to take them.
This removes the behavioral element completely. You are essentially getting a vaccine against heart disease.
That is the moment where I feel the ground shifting. We aren't just fixing broken, rare things. We are upgrading the standard human operating system to be resistant to the #1 killer in the world.
And that is just the beginning. If we look at where the architecture of these nanites is going, it gets even more sophisticated. We are moving toward a modular design.
Right, the tri-mode nanite. This part of the notes felt like we were reading a schematic for a robot.
It essentially is. The vision, and researchers are already building these components separately, is to combine 3 specific operating modes into a single therapeutic package.
Let's run through them for everyone. Mode 1 is seek and destroy.
This is primarily for oncology, cancer. The biggest problem with cancer is that it's you. It is your own cells. So the immune system has a hard time spotting it. And as we said, chemo kills everything. But a programmable nanite in seek and destroy mode is different. It carries a kill switch.
It has a target list.
Yes. It identifies specific tumor markers. genetic signatures or surface proteins that only the cancer cells have. It enters the cell, verifies the signature like checking an ID card, and triggers apoptosis.
Apoptosis.
Programmed cell death. Every cell has a self-destruct button hidden in its code. It is a safety feature. If a cell gets too damaged, it's supposed to kill itself to protect the organism. Cancer disables this button. The nanite goes in and pushes that button manually.
So the cell just deletes itself.
It packs itself up neatly and shuts down. It shrinks and is absorbed by the body. No inflammation, no mess.
That sounds incredibly clean compared to surgery.
It is. And this links to the concept of oncolytic viruses. These are viruses engineered to infect only tumor cells, and when they do, they don't just kill the cell, they make it explode in a way that alerts the immune system.
Oh, so it's like lighting a flare.
Exactly. Cancer tries to stay cold and invisible to the immune system. These viruses turn the tumor hot. They say, hey everyone, the enemy is right here. So you get a double whammy. The virus kills the tumor, and it wakes up your own immune system to finish the job.
Amazing. Okay, mode 2, restore mode.
This is for when you don't need to kill something, but you need to fix damage. Think spinal cord injuries, heart attacks that leave scar tissue, cirrhosis of the liver.
This is the regeneration aspect.
Right. The nanite delivers instructions to activate stem cells or growth factors in the local area. It says don't just scar over, rebuild. It is trying to convince the tissue to return to a factory new state.
And finally, mode 3, maybe the most important one, self-destruct mode.
This is the safety layer. You don't want these machines running forever. You don't want them drifting into tissues they shouldn't be in. You don't want a heart repair virus drifting into your brain.
So how do you stop it?
You code in a sunset clause. Once the mission is done, or after a certain number of replication cycles, the nanite dissolves. Or it becomes susceptible to a specific benign chemical. You take a pill, an antibiotic, or a trigger drug, and it wipes all nanites out.
It just ghosts.
It degrades, no trace left behind. This prevents runaway effects or long-term immune reactions.
It is incredible to think of these 3 things, killing the bad, growing the good, and cleaning up the mess all packed into a microscopic package. But the sources took a detour into something that I frankly didn't expect. Physics.
Ah, yes.
Oh.
Photonic control.
Light. Using light to control biology. This sounded fascinating, but also really abstract. Help me visualize this.
It is the frontier of the frontier. It is called optogenetics.
Optogenetics.
Basically, there are proteins in nature, like in algae or in the eyes of insects, that change their shape when they get hit by a specific color of light. Scientists have figured out how to splice those light-sensitive switches into other proteins like neurons.
So you can turn a cell on or off with a laser.
Exactly. Imagine a neuron in the brain that controls a tremor in Parkinson's disease. If you infect that neuron with a light-sensitive nanite, you could potentially shine a specific frequency of light even through the skull using infrared and mute that neuron.
No electrodes, no wires, just light.
Just light. But the really mind-bending application mentioned in the texts is about prions.
Prions are terrifying. That is mad cow disease, right?
Yes, and Creutzfeldt-Jakob disease. Prions are unique because they aren't bacteria and they aren't viruses. They are just proteins. But they are folded into the wrong shape.
It is a geometry problem.
It is. Think of a protein like an origami crane. A prion is like that same piece of paper crumpled into a tight, jagged ball. And because they are chemically the same as the healthy protein, your body doesn't attack them. But the wrong shape is contagious. If the crumpled ball hits a crane, the crane crumples too. It is a structural cascade failure.
Like a domino effect. And drugs can't stop it, because drugs look for chemistry, not shape.
Exactly. So the theory here is that since this is a structural problem, maybe we can solve it with physics, photonic bandgap structures. The idea is to use specific frequencies of light to resonate with the protein. You vibrate it, you add energy until you destabilize that wrong shape.
You shake the crumpled ball until it opens up.
In theory, yes. You are fixing the geometry of the molecule with light. It is a completely different way of thinking about medicine. It is not chemistry. It is structural engineering at the atomic scale.
This brings us to a transition point in our deep dive. We have talked about fixing things. We have talked about curing cancer, fixing hearts, untying prions. That is all restore mode. That is bringing a sick person back to normal. But the sources push further. They ask if you have a platform that can write code into the human body. Why stop at fixing? Why not upgrade?
This is the shift from therapy to enhancement, and the source text uses a very pop culture analogy here. The Predator.
The movie monster.
Yes. In the movies, the predator species travels the galaxy hunting the strongest prey so it can absorb their traits and become stronger. It is gruesome.
It is very messy.
Very messy. But the core idea, absorbing superior biological traits from other species, is actually possible through synthetic biology. But we don't need to hunt. We just need to read.
Right. The expert notes call this translation, not transplantation. And I think this is a really important distinction because people get weirded out here.
They do. People hear animal traits and they think of the fly. They think we are going to graft a salamander leg onto a human. That is transplantation. That is crude. That is 19th century thinking.
And we aren't doing that.
No, we are doing translation. We look at the salamander. We find the specific gene sequence that allows it to regrow a limb. We read that code, then we rewrite it. We translate it into human. We optimize the code so it works with our cellular machinery but keeps the function.
It is like taking a poem written in French and translating it into English. You keep the meaning, but you change the words.
Beautifully put. And because we are synthesizing the DNA from scratch in a lab, we aren't actually putting animal parts into people. We are putting in data that was inspired by animals.
So let's look at the library. What books are we checking out here? The sources list some superpowers found in nature that we could technically borrow. The first one is a tiny little tank called the tardigrade.
The water bear. These things are legendary in biology. They are microscopic. They look like little chubby bears with 8 legs. You can boil them, freeze them, dry them out into a crisp for 10 years, rehydrate them, and they walk away. But the most impressive thing, you can shoot them into the vacuum of space and bombard them with radiation.
And they survive. How?
We do know why. It is a protein called DSUP.
Damage suppressor.
Exactly, DSUP. It is a protein that physically hugs the DNA strands. It acts like a shield. When radiation hits the cell, usually it acts like a bullet. It shatters the DNA helix. But with DSA, the protein takes the hit. It absorbs the energy so the DNA doesn't break.
That is incredibly cool. There's a force field for your genome.
And here is the proof of concept. An MIT study from 2025 mentioned in the source. They didn't just study it, they used it. They delivered the instructions for DSUP into human cells using mRNA, just like the COVID vaccine technology.
And did it work?
It did. It provided temporary protection against radiation. The human cells with DSUP had significantly less DNA damage than the ones without.
Temporary is key there, right?
Yes, and that is actually a benefit. Imagine a cancer patient undergoing radiation therapy. You could give them a DSUP injection before treatment. It shields their healthy cells, their skin, their gut for a few hours while the radiation kills the tumor. Then the DSUP fades away.
Or astronauts.
Right. Deep space travel. The biggest barrier to Mars is radiation. A solar flare could cook the crew. But if you have DSUP on board, you take your shot. You weather the storm, it opens up the solar system to us.
Incredible. Okay, next trait. Regeneration. We talked about this a bit, but let's go deeper. The salamander. The axolotl.
The kings of regrowth. You cut off an axolotl's leg and it grows back. Bone, muscle, nerve, skin, perfect replica.
How does it do that? Because if I lose an arm, I just get a stump.
It forms something called a blastema. It is a lump of stem cells that forms at the injury site. It is like the body suddenly remembers how it was built in the womb. It hits replay on the development code just for that limb.
No, we can't do this.
Well, this is where the new research is shocking. A study from LSU suggests that we can, or at least we have the software for it. The genes for this kind of regeneration are likely sitting in our DNA right now.
Dormant.
Dormant. Silenced by evolution.
Why? Why would evolution turn off a superpower?
Think about our history. We are warm-blooded land mammals. If a primitive human broke a leg or lost a hand and they had to lay still for 3 months to regrow it, they would starve. Or they would get eaten by a lion. Speed was more important than perfection. So we evolved to scar over fast. Scarring is a survival strategy. It stops the bleeding, seals the wound, and lets you run away.
But we don't have lions anymore. We have car accidents. We have war injuries.
Exactly. The survival pressure has changed. So the upgrade here wouldn't be adding new genes from a salamander. It would be using a nanite to go in and flip those sleeping switches back on to wake up the blastema.
And there was a Harvard study about adrenaline that connects here.
Yes. This was fascinating. They found that in salamanders, the nervous system plays a huge role. Adrenaline signaling triggers a whole body memory of the injury. It primes the system to rebuild. It suggests that regeneration isn't just local, it is a systemic state. You have to tell the whole body we are in build mode.
It is mind-blowing to think that the ability to regrow an arm might be hiding inside us right now, just waiting for the right password.
It redefines potential.
Let's hit a couple more quick ones. Night vision.
The owl and the deep-sea fish. They have specialized opsins retinal proteins that catch infrared or low-light photons. We could code those into a human retina you would see in the dark.
That would change everything from military operations to walking the dog at night. And finally, oxygen efficiency, the seal.
Diving mammals have 30 times more myoglobin in their muscles than we do. Myoglobin is the molecule that holds oxygen in the muscle tissue. That is why a seal can hold its breath for an hour and hunt.
So if we tweaked our myoglobin levels,
You could hold your breath for 20 minutes, but more importantly, think about medical resilience. If you have a heart attack, the damage happens because the heart muscle runs out of oxygen. If your heart was packed with myoglobin like a seal's, it could survive the blockage for much longer without permanent damage.
it sounds super heroic, but when you break it down to proteins and genes, it just sounds like engineering.
It is engineering,
Uh-huh.
but that brings us to the oops factor.
Yeah. Section 6, the buzzkill section. But a necessary one, because if engineering has taught us anything, it is that code has bugs. And biology is the most complex code there is.
It is the risk of playing God argument, but stripped of the theology and replaced with practical safety concerns. What happens if the code bugs out?
Technical risks. Let's start with off-target edits.
This is the Control-F error. You send the nanite to cut gene A, but maybe gene B looks a lot like gene A. The nanite gets confused and cuts gene B.
And if gene B is important.
If gene B is a tumor suppressor, you just gave the patient cancer while trying to cure their blindness. That is the nightmare scenario.
Or runaway expression.
You tell the body to grow more muscle like the seal, but you don't calibrate the stop signal correctly. You get muscle growth that creates too much mass. It could crush your bones or strain your heart. Biology is all about balance.
Or the immune system just freaks out.
That is a major one. We talked about AAVs being stealthy, but they aren't invisible. Some people have natural immunity to them. If you inject a high dose, the body might launch a massive cytokine storm. That can be fatal.
So it is not as simple as downloading an app. You have to navigate the body's defenses.
No, it's not. But the risks aren't just biological, they are sociological. And this is the one that keeps me up at night, the class divide.
The cost. We mentioned Casgevy earlier.
Casgevy costs about $2.2 million per patient.
$2.2 million. Most insurance companies balk at that. Most countries can't afford that. Now, we are imparting this directly from the source materials, which present a really stark warning here about economics. Both left and right-leaning analyses of this point to the same massive problem.
Exactly. So imagine we unlock these upgrades: immunity to cancer, higher intelligence, better physical resilience. Who gets them?
The wealthy.
Yeah.
The people who can afford a $2 million procedure.
And everyone else stays baseline. We risk creating a biological split, a literal speciation event where the rich are genetically superior to the poor, not just better educated or better fed, but biologically better.
That is terrifying. It is bad enough with money, but if it is built into your DNA, if your children are born better because you paid for the premium package?
It creates a permanent underclass. That is why the source material emphasizes that enhancement policy must be treated as a global commons problem. It cannot be a luxury market. It has to be accessible, or it will tear society apart.
And there's another line in the sand. The difference between editing me and editing my kids.
Somatic versus germline. This is the golden rule of bioethics right now.
Explain the difference.
Somatic editing means editing the cells of your body. Your liver, your eyes, your muscles. If you get an upgrade to your eyes and then you have children, your children do not inherit that upgrade. It dies with you.
Okay. That feels safer. It is contained.
It is. It is consent-based. You chose it for yourself. But germline editing means editing the sperm, the egg, or the embryo. That change is copied into every cell of the resulting human. And crucially, it is passed down to their children and their grandchildren.
You are editing the lineage. You are editing the future of the species.
You are. And that future child cannot consent. That is why the scientific community came down so hard on He Jiankui in 2018 when he edited those embryos in China. It was a violation of the future. We don't know enough about the long-term effects to be writing code that lasts for generations.
So the sources suggest a middle ground here.
Yes, the reversibility clause. The idea is that we should focus on temporary enhancements first, like the mRNA DSUP for radiation. It wears off. It is not a permanent tattoo on your soul. It is a raincoat you put on and take off.
That makes a lot of sense. And we have safeguards in place now, right? It is not the Wild West.
We do. The FDA requires 15 years of surveillance for anyone who gets a CRISPR therapy. We are watching these first patients like hawks. And every nanite design now includes those kill switches we mentioned earlier. We are proceeding with caution.
It seems like we are walking a tightrope. On one side, the end of disease and the beginning of a new kind of human potential. On the other side, errors, inequality, and ethical nightmares.
That is the nature of powerful technology. Fire cooks your food, but it burns your house down. This is just a very, very sophisticated fire.
So as we wrap up this deep dive, I want to go back to the image the expert nuts ended with, the quill and the code.
It is a poetic image. For all of human history, biology has been destiny. You get what you get. You get your parents' genes, you endure disease, you age, you die. We were the readers of our own story.
But now,
Now we are picking up the pen. We are becoming co-authors. We aren't just enduring nature, we are directing it. Evolution used to be blind, random, and slow. Now it can be intentional and fast.
it changes the definition of what it means to be human. If the body is a canvas, what do we paint?
That is the question. And it is not a question for scientists alone. It is a question for everyone.
And that is where I want to leave you listening today. We have talked about curing cancer, regrowing limbs, seeing in the dark. But let's make it personal. If you could pick up that quill, that viral nanite, and write one single upgrade into your own DNA, what would it be? Would you want to live longer? Would you want to never feel pain? Or would you be afraid to touch the code at all?
And are we ready for the responsibility of being the writers?
That is the big one. Thanks for diving deep with us. We will see you next time.